Okay, let's begin. For a variety of reasons, most of which I talked about a little bit last time, I think it's really dubious that neuroscientific evidence, that is, evidence about biological structure and function, can actually make a contribution to theory at the psychological level of analysis. That is, knowledge about how the brain works isn't really going to tell us anything about how the mind works. However, if what you want to know is how the brain does it, that is, how mental structures and processes are represented and implemented in brain tissue, well, neuroscience is the way to go, okay? The doctrine of modularity, which achieved prominence in cognitive neuroscience, has also guided research in social neuroscience. And various what's now called social neuroscientists have done studies of brain damaged patients and also brain imaging studies of intact subjects to try to identify what modules might be present in the brain that are particularly active in processing social information. Now again, if you just think about it for a second, you can't just image somebody's brain and hope to stumble upon a module that's dedicated to social cognition. You have to have some sense of what you're looking for before you do the imaging study, because what you have to do is to design a particular task that is going to elicit a particular kind of processing activity from your subject, so that you can identify what brain regions are active when the person engages in that processing activity. So in fact, the first step towards identifying brain modules or systems or brain systems that are particularly dedicated to social tasks is to try to figure out what those modules might be, just kind of get some sense of what you're looking at. And along the way, as I described all too hurriedly at the end of the last lecture, there have been a number of proposals along these lines. My favorite comes from Ray Jackendorf, who's a linguist at Tufts University, who's argued for a variety of reasons for a particular package of modules for social cognition. So, for example, he's suggested that we have dedicated brain modules for face recognition, for voice recognition, for the detection of affect, and for the detection of intentionality. And he does that simply by arguing that these are specialized kind of stimulus events, and they probably require specialized capacities for dealing with certain kinds of input. He's also argued that there might be a brain module that distinguishes between animate and inanimate objects, and another brain module that handles the proper names of objects, calling somebody John instead of that guy over there. And he makes this argument based on what he calls developmental priority. It turns out that little kids make the distinction between animate and inanimate objects, things that move on their own versus things that have to be moved, very, very, very early in life. And he suggests it happens so early that there might actually be a brain module for that. And also, kids start using proper names very, very, very early in life. And he suggests that there might be something brainy about that. Based on anthropological studies, he's argued that there are certain cultural parameters that are universal. That is, that they're found in certain features that are found in every culture. So, for example, every culture has categories of kinship. Now, as we discussed earlier in the course, different cultures have different categories, but everybody recognizes the difference between kin and not kin. The distinction between in-group and out-group seems to be universal across cultures. Social dominance relations and things like that. And Jackendorf has speculated that precisely because these kinds of categories, these kinds of distinctions are universal across cultures, they're precisely the kinds of things that if anything's going to be represented in the brain, something that's universal is going to be represented in the brain. So Jack and Doff has basically suggested that these are the things we would want to look for. We'd want to find, for example, we might want to look for a module that is specifically geared towards recognizing faces as opposed to other kinds of visual objects. Another module that's specifically involved in recognizing voices as opposed to other kinds of auditory stimuli. Was that a question? Yeah. Yeah. Jack and Doff does not get into that question at all. He's a linguist. He's not a brain stabber or a brain imager or anything like that. He just argues that if you're going to find dedicated social modules in the brain, you're going to see something like that. But remember that Jack and Doff is a linguist. And in the study of language, we've already identified both modules, or something module-like, and systems that are involved in language processing. So, for example, we have Broca's area in the interior portions of the frontal lobe, and we have Wernicke's area around the posterior portion of the temporal lobe, and we know that those areas are specialized for language. Broca's area for speech, production, and Wernicke's area for, to make a long story short, the processing of meaning. And we also know that there's a huge bundle of fibers that connects those two areas. Okay? So that if I put up a word here and ask you to pronounce that word, the first thing we'll see is activation in your visual cortex as you read the word. Then activation is passed to Wernicke's area as you comprehend the word, and then activation is passed to Broca's area as you utter the word. So there's a kind of system there. There are modules that are dedicated for specific things, but they work together as a system, and that's really what Jack and Doff has in mind here. That's his analogy. Okay, he says if we're going to have these things, they're going to kind of sort of work like the ones that we have in visual perception and the ones that we have in language processing. And so what you have to do, as you'll see in a second, is once you've got an idea about what might be modular, okay, then you can design a task that will help you to identify that module in the brain by sticking somebody in a magnet and asking them to perform a particular task. I mean, that's what you do, you know. You stick them in a magnet, you ask them to perform a task, and you see what parts of the brain light up when they do that as opposed to some kind of control condition. Or you find somebody with a particular form of brain damage and you ask them to perform a particular kind of task like recognize faces or recognize voices, and you find out that they can't do that, and you say, well, look, they've got damage in this particular place, they can't perform this particular task, therefore the module that performs this particular task is likely to be located at that particular place. That's the logic that you use, okay? But the essential point here is that you gotta know what you're looking for before you're ever gonna have any chance of finding it. If you can't just put somebody in a magnet and say, think about your mother, and expect to find the mother area in the brain. At the very least, you're going to have a control condition where you say, think about your aunt, or think about Nancy Reagan, or something like that, okay? So that you can get some differential pattern of activation. So let's see how this kind of thing gets implemented in practice. In fact, neuroscientists of various stripes have begun to identify brain areas that appear to be modular in just the way that Jack and Doff and Fodor suggested. Here, for example, are some images from the work of Nancy Canwisher and her colleagues, who believe they've identified what they call a fusiform face area, an area in the fusiform gyrus right there, the lower portion of the temporal lobe. That seems to be involved in the recognition of specific faces. Here is another area, what she calls the temporal parietal junction mind area, right there at the junction of the parietal lobes. That seems to be activated when people make inferences about other people's mental states, what they want, what they feel, what they're thinking. And here is yet a third area, what she calls the extrastriate, the body area, next to the striate cortex that's involved in vision, and that seems to be activated whenever people try to perform a task involving their own bodies. If you ask somebody to, where's your right arm right now? And they perform that task, you see this area of the brain being activated. These are three very early studies that seem to identify brain modules that are involved in more or less specific social or personality type tasks and not seem to be activated under other conditions. Notice the dates, 1997, 2001. This is all relatively recent material. But Canwisher and her colleagues, plus the arguments about modularity that really came to us from cognitive neuroscience, set off a flood of research where as soon as social psychologists could get hold of magnet time, they threw their subjects in magnets and asked them to perform various kinds of tasks, the result of which was that by 2007, in a chapter in the Annual Review of Psychology, Matt Lieberman, who is a pioneering social neuroscientist, was able to identify fully 21 separate modules in the brain, that is 21 separate areas of the brain, each of which seem to be specifically associated with some particular task. So back here we have the temporal parietal theory of mind area that I talked about before. Here we have an area that seems to be involved in processing information about the self, a top problem I'm going to talk about in a minute, and so on and so forth. We don't have to get into the details of this. The point is that this is progress of a sort. You've now, that's 2007. There have been about half a dozen similar kinds of areas identified since then. They're just now represented on this cool little map. So that suggests that really there is something here that you can do. You can identify, just as we identify specific pieces of the brain that are involved in visual perception or language processing or memory in general, we can identify particular parts of the brain that are implicated in social processing as well. Well, not so fast, okay? This is a project that can work, but it's also a project that is fraught with difficulties, not the least of which is that you have to have the right kind of experiment to do these kinds of studies. If you don't have the right task, if you don't have the right control group, put another way, if you haven't properly analyzed the task you're asking your subject to perform at the psychological level of analysis, putting the subject in a magnet and asking them to perform that task and then trying to interpret the neuroimaging results, that's not going to get you anywhere at all. Let me give you two examples of this. The first one comes from an enormous project engaged in by a large number of investigators now, searching for a particular brain module that is involved in self-referent processing. That is, some part of the brain that is activated whenever you think about yourself. It makes sense that there would be such an area of the brain, okay? The distinction between self and others is universal. Everybody makes that distinction. People with borderline personality disorder have more difficulty with it than other people do, but everybody still makes that distinction. So it makes sense that we might be able to find a module in the brain, some piece of the brain that is always activated whenever people think about themselves. Well, the first study of this sort was performed by Fergus Craik, a Canadian psychologist, and his colleagues in the late 1990s, and their task was based on a phenomenon in memory that's known as the self-reference effect. It turns out that when you present words to subjects and ask them to remember them, they can remember them. But if you present words to subjects and ask them to relate the word to themselves, they remember them really well, okay? So if I presented you with a set of trait adjectives and said, is this trait characteristic of you? You just say yes or no, yes or no, yes or no, as we go through the list. And then later on, I surprise you with a test of memory. You'll remember those trait adjectives very well, much better than you would if you were asked to perform some other kind of test. So here's the general format for a self-reference effect experiment. You present subjects with words, typically trait adjectives, like the word extrovert, and you might ask the subject to perform what's known as an orthographic task, making a judgment about the visual appearance of the word. Does the word extrovert have an E in it? Or you could ask them to perform what's known as a phonemic task, making a judgment about the sound of the word. Does extrovert rhyme with pervert? Okay, that's the question they use. Semantic orienting test asks subjects to make a judgment about the meaning of the word. Are extroverts happy? Okay, and the self-referent judgment asks people to make a judgment about themselves with respect to the word. Are you an extrovert? Craik and his colleagues were relying on research by Timothy Rogers, another Canadian psychologist, and his colleagues done long ago now, that first identified the self-reference effect in memory. Here we're plotting the probability of recall as a function of the various orienting tasks, and you can see this is usually what's known as the levels of processing effect. Semantic processing gives rise to better memory than phonemic or orthographic processing. That's a very familiar effect in memory. But what Rogers and his colleagues discovered was that self-referent processing gave rise to an even bigger effect. Now, the explanation of this level of processing effect basically says that what gives these words an advantage in memory is that they make contact with fairly large pre-existing knowledge structures. In the orthographic or phonemic task, the items don't make contact with a lot of pre-existing knowledge. In the semantic task, they do. Well, look at that memory advantage, leading Rogers and his colleagues to conclude that the self was by far the largest knowledge structure stored in memory. So what Craik basically did was to repeat the Rogers experiment with subjects in an imaging machine. This is PET imaging, actually, not fMRI. And basically tried to identify an area of the brain that was activated specifically when subjects made self-referent judgments about words. As I said, they used PET imaging, and they had a couple of different conditions here. Here's the critical one, describing self. Now, the obvious the control here is making the judgment about whether a trait describes some other person, and so that they could have the same person for everybody. Remember, this is a Canadian study. They asked people if the trait adjectives described Brian Mulroney, who was then the Prime Minister of Canada. There were a couple of other tasks as well. Use a standard subtraction methodology that's used in these kinds of studies to identify a part of the brain that is activated specifically when subjects perform self-reference judgments. Well, that didn't work out so well. What you can see here is that if you compare parts of the brain that are activated in self-reference processing versus making a judgment about syllables, there are some areas, but the same areas are activated when people make an other referent kind of judgment. Same kind of thing here. Okay. The point here is that when they use their standard statistical ways of analyzing this data, they didn't get much of anything except maybe a little bit of increase in activation in an area of the brain known as the right anterior cingulate cortex, a piece of the brain that's embedded in the medial portion of the frontal lobe. Fortunately, there was another statistical technique available to them. It's a little bit more sensitive. You don't have to know these details. God love you if you do. But what using this allegedly more sensitive statistical technique, they were able to identify a couple of parts of the brain that were more activated when people made self-referent judgments than when they made other referent judgments or the other kinds of judgments. A portion of the medial frontal lobe in the right hemisphere, a portion of the frontal gyrus on the lateral portion of the right hemisphere, and a part of the right inferior frontal gyrus. Here are the areas that they looked at. Here you have this is the left hemisphere. I know that, but that's the diagram I've got. Okay. Here's the inferior the gyrus of the frontal lobe. Here's the middle gyrus of the frontal lobe. It's these areas: Brodmann's area 45, 10, 46, and in the medial portion of the frontal lobe, Brodmann areas 9 and 10. Those are the areas that seem to be associated with self-reference. So they published this paper. That we found it. We found the cells in the brain. Here's a particular portion of the brain that's activated when people make self-reference judgments. Okay. The problem is that as people discovered later on, that self-referent judgments don't provide a unique advantage in memory. That other referent judgments also provide a unique advantage in memory, provided that the subject knows the other person well. And the problem here is that in the Craik experiment, they made a mistake, basically, which was to compare self-referent judgments against an other referent judgment where the other person being judged was somebody that people just didn't know very well. I mean, you might think you know your prime minister well if you're in Canada, but you don't know him as well as you know your brother or your roommate or your girlfriend or something like that. The Craik experiment was an important pioneering experiment, but it had this flaw, which was that the other referent judgment was not about a person that the subjects knew particularly well. And it turns out that when you clean up that problem, you ask subjects to make judgments about themselves, and you ask those same subjects to make judgments about somebody else that they know well, their brother, their sister, their roommate, their mother, somebody like that. At that point, the specific self area begins to disappear. Kevin Oxner, then at Columbia University, and Jennifer Beer, Berkeley PhD. This was part of Jennifer's graduate work here, in fact, found that exactly the same areas of the brain were activated when people made self-referent judgments, the same portion of the temporal lobe that Craik had, of the frontal lobe that Craik had identified. That's also activated when people make other referent judgments. So it's possible that this is an area that's involved in personality judgment, but it is not an area that appears to be involved in specific self-referent judgment. The self is still somehow lost in the brain, and we haven't quite found it yet. So when you do the study right, when you have a proper control condition where you can control self and other judgments for familiarity, likability, things like that, the area that is apparently specifically associated with self-reference disappears. Okay. Now you don't have to believe just that one study. Okay. But again, I mean, here it is. This particular area is what Oxner and Beer found was activated when people made self-referent judgments, but was also activated when people made judgments about a close other person. So it does not appear to be an area that's specifically associated with the self. Martha Farah and her colleagues did a review of this literature a couple of years ago, in which they looked at, tried to use tests that would identify the physical self, recognizing your own picture as opposed to the picture of somebody that's close to you, the psychological self, making judgments about traits or autobiographical memories or whatever, and they concluded on the basis of their review of this literature that there were no areas of the brain that were specifically activated when people made self-referent judgments, when the control condition was an other reference judgment where the other person was well known. Here's face recognition, for example. There are some areas. This is essentially where the fusiform, the face area is, and that's activated when you recognize your own face, and it's also activated when you recognize the face of somebody that you know well. Here is when you're looking at self as an agent engaging in some activity, or another person as an agent engaging in some activity. You get activation here, but you get the same activation when you're looking at self or other. And for trait ratings, again, the same kind of thing. Here you've got this portion of the temporal lobe that we've looked at before, and it is activated both when people make self-referent judgments and judgments about other people. Leading us to conclude, at least for now, that as far as the brain's concerned, the self isn't special. As far as the brain is concerned, the self is just another person. It's a person you might know particularly well. It's a person you might like particularly well. But as far as the brain appears to be concerned, the same parts of the brain are involved in making judgments about the self as are made about other kinds of people. So again, this is an illustration of just how careful you have to be in doing these brain imaging experiments. If you don't have the right task, if you haven't analyzed the task properly at the psychological level of analysis, the neuroimaging results are going to be misleading, if not downright wrong. Here's another example of this, which is the area of the brain that appears to be associated with processing faces. In the same way that it makes a great deal of sense conceptually that there would be a part of the brain that's dedicated to processing information about the self, it also makes sense that if there's a special social module in the brain for anything, it would be a social module in the brain for face perception and face recognition. The face is, after all, the primary social stimulus. It's the point of contact between infant and caregiver. It's the channel by which we get information about other people's identities and by which we get information about their emotional states and all of that kind of stuff. If there's anything social that deserves a dedicated module in the brain, it's the face. Okay, that's really all that has to be said about that. If there isn't a face module in the brain, there's probably not any other kind of social module. And as I said, we're going to skip this. As I said before, there does appear to be, Can Wisher and her colleagues appear to have identified a particular area of the brain that seems to be dedicated to the processing of this primary social stimulus, the face. Some evidence along these lines is neuropsychological evidence and comes from cases of brain damage, and especially a particular form of brain damage known as visual object agnosia. Agnosias really are failures of semantic memory, if you will. A person with visual object agnosia can describe an object that's presented to him or her visually, but they can't name the object, they can't identify the object, they don't recognize the object as familiar, and they typically can't demonstrate how the object is going to be used. visual object agnosia is nicely characterized as percepts that are just percepts. They're percepts that have been stripped of meaning, which is why we call it an agnosia. Well, it turns out that there's a particular form of visual object agnosia which is quite interesting to social psychologists, and it's known as prosopagnosia, a neurological condition that was first described by Walter Botimer in 1947. And this appears to involve a specific deficit in recognizing faces. These patients have the ability to, they can certainly see the face. You know, if you put a face in front of them and say, is there an object there? They'll say yes. If you ask them to describe the face, they can do that. If it's a color photograph, they can say, well, the person has blue eyes, and the person has red hair, and the person has freckles, and all these kinds of things. However, what they can't do is they can't put a name to the face. They can't tell you who the person is, even if the face belongs to a person who ought to be familiar to them. And there are some cases of this where the difficulty of recognizing faces occurs in the context of visual object agnosia in general, which is they can't recognize anything else either. But there are, what Botimer was interested in were some cases where there's a pure deficit in facial recognition. The person has no ability to identify familiar faces, that is, faces that should be familiar, doesn't have a feeling of familiarity, but can recognize other kinds of objects. And what Botimer discovered on autopsy, you let these patients die and then you look at their brains, is that, well, you don't let them die, right? Everybody dies, right? You don't kill them for this purpose. You just let them die, you wait for them to die, and then you get hold of their brains. And what Botimer discovered was that these patients who had a pure prosopagnosia, pure inability to recognize faces, had damage to a particular area, basically at the junction of the temporal and occipital lobes. Brodmann's areas 18, 19, and 37. There's 37, there's 18, there's 19. Damage is right in there, okay? Leading some individuals to suggest that this is a face area, a part of the brain that is specifically geared to recognizing faces. And later research has shown that even with neurologically intact subjects, if you ask subjects to identify faces as opposed to other kinds of objects, you'll see activation in this area as well. It was that kind of evidence, in fact, that led Candlisher and her colleagues to identify this area as what's known as the fusiform gyrus, as the fusiform face area. As with the self, however, this turns out to be not quite as easy as you'd like it to be. Here's another depiction of where this area is. Here's the occipital lobe. Here's the temporal lobe. There's a little notch here, okay? And the fusiform area is basically here. There's area 30, Brodmann's area 37, 18, and 19. So, and it kind of makes sense that that's where you'd find an area for recognizing faces, because as you know, the occipital cortex has a lot to do with visual processing. Temporal lobe has a lot to do with memory processing. Recognizing faces, got to connect up what you see with what you remember, and it kind of makes sense that if there were going to be a face area, you'd find it there. And as I say, because if there's any module in the brain for any kind of social task, there ought to be one for face recognition. The idea that this is a face area, a part of the brain that's specifically dedicated to identifying faces, makes a great deal of sense and was kind of quickly and almost universally accepted as the identification of a particular social module. Yes.
Some people say, like, I never forget a face.
Yeah.
They have in a large
Oh, yeah. Well, that might be the case, okay, but you have to be careful. People with, we know that the hypothalamus regulates eating, but people with eating disorders don't have damaged hypothalami. They don't have shrunken or expanded hypothalami. And in general, it's not easy to correlate brain size with ability. For example, Albert Einstein's brain looks just like everybody else's brain. We know that because UC Berkeley's own Marion Diamond did the histology on Einstein's brain, and what she found was that he had a lot more glial cells than average, but not a lot more neurons. We do know that when people get very good at something, the anatomy of the brain changes in certain respects, okay? So if you become a really good violin player, the areas of the brain that especially mediate the left, the finger area, they get bigger. Not more neurons, but more connections, okay? So it might make sense that if there really were people who never forget a face, and they really don't, okay, we might find that this particular area was larger. That's a perfectly reasonable hypothesis. To my knowledge, unless somebody wants to correct me, which is okay, to my knowledge, that study has never been done. But you might also find it someplace else. For example, we know, or we think we know, almost everybody agrees, that memories aren't located in any particular place. So it's not as if the person has all those memories stored there and it just has to get big like your filing cabinet in your desk. But if you're particularly good at something, that part of the area might be better. But it turns out that the issue turns out to be much more complicated than that, in part because, again, there are difficulties with the demonstration that prosopagnosia involves a specific deficit in recognizing faces. And the problem here is kind of subtle, but it looks like this. What Bottomer would do would be to present one of his patients with the picture of a person who should be familiar to them and say, do you know who this is? And the patients would say, no. They'd say, well, can you describe the person? Yes, he's young and he's male and he's got short cropped hair and he's got dark complexion and stuff like that. Do you know who he is? No, I don't know what he is. I don't know who he is. Then Bottomer would present a picture of a non-face object like a house and say, do you know what this is? And the person would say, it's a house. Now, if you think about it for a little bit, those are two very, very different questions, okay? Who is this asks you to identify the object at a specific level of categorization known as the subordinate level of categorization. It's not enough to say, oh, it's a black guy, okay? Or even, oh, he must be a government official, otherwise he wouldn't have the American flag in the background or anything like that. But the question, what is this, can be answered at what's known as the basic level of categorization, which is to say, it's a house. Just because the person can identify a house as a house, but can't identify Barack Obama as Barack Obama, doesn't mean that the person has a specific deficit in recognizing faces. You've got to control the tasks for level of categorization. So what you really want to do is to ask, you know who this is? Okay? Do you know what that is? You can only claim that there's a specific deficit in identifying familiar faces if the control task is also requires you to identify a specific object at the same level of categorization. And the question is, do prosopagnosic patients who don't identify this person correctly also fail to identify that object correctly, to say, that's the White House, okay? Well, it turns out that many prosopagnosia patients can't identify the White House as the White House. Okay? So it doesn't look at like a particularly specific area that's geared towards face identification. It looks more like this area of the cortex around the so-called fusiform area seems to be involved in identifying objects of any sort, whether they're faces or people or houses or whatever, at a particular level of categorization, at the subordinate level of categorization that involves identifying specific instances. The person who noticed this, by the way, just so you have some inspiration for those of you who are going on to graduate school, or those of you who are in graduate school, was a graduate student, Isabelle Gautier, who's now a professor at Vanderbilt University, who got interested in, she's a vision scientist, a cognitive neuroscientist, who got interested in this problem, and all of a sudden she just kind of noticed this one day, that there was this problem with these tasks, and then set off for her doctoral dissertation to correct the issue. It was Gautier, in fact, who discovered that many prosopagnosic patients can't identify objects, non-face objects, at the subordinate level of classification either. But most of her work, let's do this, right, most of her work had to do with imaging the brains of people as they perform particular kinds of tasks. Most of these studies were PET imaging studies. Some of them were fMRI studies. That detail doesn't matter. But in one of her early studies, for example, she took subjects and asked them, showed them pictures of various kinds, and asked them to make categorization judgments at two different levels of categorization. Either the basic level of categorization, asking them, putting up a picture of a bird and saying, is this a bird or not? Or a subordinate level of categorization, where you put up a picture of a bird and you ask the subject, is this a pelican or not? And what she discovered was that this task, which is not a social task, doesn't involve face stimuli or anything else that's particularly social, also recruits the fusiform area, okay? That when you make judgments about objects at the subordinate level of categorization, that task activates the fusiform area, regardless of whether you are recognizing faces or you're recognizing other kinds of objects. In another series of studies, she actually took college students who were not experts in a particular area, and then over a long period of time, she actually hired them as work study students, taught them to become experts in classifying various kinds of objects. Classifying cars, is this a car? Is this a Buick Riviera? Okay. Birds, is this a bird or is it a pelican? She also trained some subjects to become experts in classifying snowflakes. It turns out snowflakes come in 7 kinds. You didn't know that, did you? Okay. Snowflakes come in 7 kinds. There are 7 broad categories of snowflakes. And if you really work at it, you can become an expert in classifying snowflakes, so that when you see something like this, you can tell what kind of snowflake it is. She taught other subjects to classify fingerprints, okay, to recognize individual fingerprints at the subordinate level of categorization. And she also taught subjects to recognize wholly artificial objects known as greebles. These were objects that were developed in the laboratory of her dissertation advisor, Mike Tarr. They're kind of sort of face-like, but not really. But the really nice thing about these greebles is that they come in what are known as races, genders, and ages, depending on things like whether the body is fat or thin, or whether the ears go up or down, or whether the nose is long or short, or whatever. But what she did was to teach subjects to classify these objects that all pretty much look the same to us, but after a couple of months of training, you become very, very, very good at this, okay? And what she discovered was that as subjects became experts in categorizing these non-social objects, fingerprints, snowflakes, automobiles, birds, greebles, they progressively more and more recruited this fusiform area, so that when they became experts in subordinate level classification, they used the fusiform area to make these kinds of judgments. Here's some more greebles. They're kind of cool, right? And you just kind of look at them, you kind of get a sense that they come in different types, and they do. I'm not a greeble expert, so I can't tell you what they are. Body shape, okay, and then the orientation of their parts, whether they go up or down, and all that. Those are the basic categories of greeble stimuli. But the point here is that there is nothing face-specific about the fusiform area. The fusiform area is activated whenever people make classification judgments at a particular level of categorization, what's known as the subordinate level of categorization, where you have to distinguish pelican from non-pelican among birds, Barack Obama from non-Barack Obama among males, male greebles versus female greebles, and so on and so forth. That's what the fusiform area seems to do. That's just more of this. Okay, there's the snowflake study. There's also a face study here where people were classifying faces as male or female or white and black. Same thing. This test obviously activates the fusiform area, but so does classifying snowflakes. Leading Isabelle Gautier and her advisor Mike Tarr to suggest that the fusiform area is not dedicated to faces, but rather is dedicated to a much broader kind of cognitive task, classifying objects at particular levels of categorization, subordinate levels of classification, pelicans, robins, sparrows, greebles of particular kinds, Buicks as opposed to automobiles, and so on, and that the fusiform area, far from being dedicated and specifically geared to recognizing faces, actually is quite flexible in terms of what it operates on. It looks like an area dedicated to, specifically dedicated to faces, because recognizing faces is a really good example of subordinate level classification. What we do when we recognize somebody's face as familiar, or we put a name to a face, or we recognize somebody's voice as familiar, arguably, or put a name to a voice, is we classify it not just as a face or a human face or whatever, but we classify it as somebody's face. Okay, the point of this is neuropsychology and neuroscience, brain imaging, studies of brain damaged patients of all sorts, can help us to identify parts of the brain that are specifically dedicated to particular social cognitive tasks. In fact, if you want to know the answer to that question, you have to do neuroimaging. That's basically what it's really good for. But before you can do the neuroimaging, you have to A, know what you're looking for. There has to be a theory that suggests to you what kinds of tasks might be performed by dedicated modules. And before you can find that dedicated module in the brain, you have to have an experimental task that actually taps that theoretical process. In this way, as in all other ways, I think, psychology drives the neuroscience, not the other way around. That's what I mean when I say that neuroscience without psychology is just neuroscience. It's a science that tells us what neurons look like and how they function chemically, but it doesn't tell us what they do. For that, we need a psychological theory. Okay, next week we'll look at the development of social cognitive functions. I'll have some things to say about autism, but I'll also have some things to say about chimpanzees. See you then. Have a good weekend.